When oxygen delivery to the human body stops, a metabolic clock begins to count down. While the central nervous system suffers rapid and irreversible damage, peripheral tissues, like skeletal muscle and skin, are more resilient. These peripheral cells possess large internal reserves of glycogen that provide a temporary emergency power supply. Using this glycogen without oxygen initiates a specific biochemical sequence that eventually destroys the cell from within. Cellular death from suffocation is not a passive fading away, but a violent self-dismantling initiated by the cell's own survival mechanisms. At 5 minutes, the cell shifts to anaerobic glycolysis. It trades glucose for minimal ATP, but creates a secondary output, lactic acid and free protons. Glucose ferments into pyruvate, then lactate. Intracellular pH drops precipitously from a healthy 7.4 down to a hostile 6.0. This state of severe acidosis changes the electrical charges on amino acid side chains within the cell. Because of these severely altered charges, critical structural proteins and metabolic enzymes rapidly unfold and lose their functional geometry. The cell's attempt to survive without oxygen is now actively dissolving its own internal biological machinery. 10 minutes in, the focus shifts to the mitochondria, which are failing under extreme oxidative stress and a heavy influx of calcium. The stress triggers the opening of the mitochondrial permeability transition pore. As this high conductance channel opens, water rushes into the hypertonic mitochondrial matrix. The inner membrane swells, unfurling its delicate folds, known as cristae, until they press against the outer boundary. The internal pressure quickly becomes unsustainable. The outer mitochondrial membrane physically ruptures, violently expelling its contents out into the cytoplasm. These ruptured mitochondria dump cytochrome C into the cell. In a healthy environment, this would initiate apoptosis, a programmed orderly cell death. However, apoptosis requires functional ATP. Because the energy grid has failed, the cell cannot execute an orderly suicide. Robbed of the energy needed for a peaceful end, the cell is condemned to oncosis, a chaotic, inflammatory, and explosive death. 15 minutes in. At the tissue level, the microcirculation has reached a point of complete stagnation. In the post-capillary venules, gravity causes erythrocytes to settle out of the plasma. They aggregate into stacked coin-like chains known as rouleaux. Inside the individual cells, the total loss of ATP activates calcium-dependent calpain proteases. These proteases sever the links between the outer plasma membrane and the underlying actin-myosin cytoskeleton. Deprived of its internal scaffolding, the plasma membrane detaches and bubbles outward, forming irregular protrusions called blebs. The cell is now swelling uncontrollably. These 20 minutes reveal a fundamental truth of our biology. Human life is not a static state, but a continuous, energy-dependent struggle against entropy.